My name is Michael Reich, and I direct software development for the lab of Jill Messerov at UCSD. I'm going to describe the features of the GenePattern notebook environment and show you how we've used it to create a collection of single-cell analysis notebooks that can be viewed, run, and adapted by the scientific community on our online notebook workspace. The electronic analysis notebook has become the de facto standard for communication among data scientists. It has taken hold in every field of science, but especially in bioinformatics. Although notebook-based systems have existed for many years, the Jupyter Notebook environment was the first to capture the wider needs of data scientists and scientific users. This is due to the unique benefits of the notebook environment. Notebooks combine the scientific narrative, including the text, graphics, and multimedia, with the code that executes the actual analysis. The notebook format allows for practically unlimited space, so the scientific process, as well as the results, can be documented without concern for page size or length limitations. Notebooks can be shared online, downloaded, and run locally or in a binder container. Notebooks offer reproducibility through providing code transparency and well-documented analysis workflows. Because of their flexibility, notebooks can be used in every phase of an experiment, from its conceptual design to the publication of results. However, for genomic and bioinformatic analyses, the notebook metaphor presents a number of challenges. The first is that notebooks assume that the user is a programmer. To use and understand notebooks, a scientist needs to have a background in a language such as Python or R, and familiarity with the packages that provide analysis and graphing functionality. Many biologists and bench researchers who could benefit from the features of notebooks are limited by their programming experience. This is especially true in cases where bioinformaticians share notebooks that are useful but require the user to modify the code to adapt it to their requirements, even if that's only to change the parameters of a function. And even the entry of formatted text is done following the programming paradigm of the markup language to express formatted text. For example, in Markdown, to create a table that looks like this, you need to write text that looks like this. And in Latex, to create mathematically formatted text like this, you need to write code that looks like this. There is also the challenge of providing access to existing bioinformatics methods. Many of these are command line tools, so you could install them onto your Jupyter environment with all of their dependencies and maintain them that way. But that prevents your notebooks from being adapted by others who may be on a different platform. Also important is that many bioinformatics analyses are compute intensive. You might want to create a workflow that includes RNA-seq analysis, non-negative matrix factorization, or other tasks that would overwhelm the compute capacity of the machine running your notebook server. This is especially true in environments that provide hosting for multiple Jupyter users through Jupyter Hub. It would be ideal to offload compute-intensive analyses to the resources best suited to support them. To summarize, what we need: easy access to existing analysis tools in a way that doesn't require ongoing maintenance of each tool. An easy way for non-programming scientists to access the methods in notebooks developed by bioinformaticians. Tools that don't require knowledge of a markup language to create richly formatted text. The easy publication and dissemination of notebooks, because it would be ideal for other users not to have to download and run a notebook locally or to use Binder or a similar online service that limits a user's ability to run and modify a notebook to what can be done in a single session. And finally, for common workflows, users shouldn't have to start with a blank page. There should be templates supporting common bioinformatics analysis scenarios that scientists can easily adapt to their own objectives. These are the requirements that led to our development of the GenePattern notebook environment. This environment gives you all of the notebook features of Jupyter and is fully compatible with Jupyter and not a separate fork of the code. The GenePattern notebook environment adds the ability to utilize analyses from any GenePattern server in addition to a number of other general features to make it easier both for non-programmers to have access to the full range of genomic analyses required for today's integrative genomics experiments, and for notebook developers to make it easier for their own notebooks to be more accessible to users at all levels of computational sophistication. Access to analysis tools is provided through the GenePattern platform. The GenePattern architecture wraps analysis methods as accessible web forms. The main GenePattern server runs on the Amazon cloud, utilizing the elastic compute capabilities of AWS to run analyses on the most suitable resources, for example, compute nodes with high memory or rapid data transfer as required. GenePattern hosts hundreds of analyses, which we add to frequently based on our scientific collaborations and user requests, providing a considerable cross-section of platform-specific methods as well as general machine learning approaches. There are also other dedicated GenePattern servers that provide access to high-performance compute resources to run especially resource-intensive algorithms. You can connect to any number of GenePattern servers from within a GenePattern notebook, combining the unique strengths of all of the publicly available resources in the GenePattern ecosystem. Let's look at the features we've added to the Jupyter Notebook system to enable this functionality. A GenePattern login cell allows users to access any available GenePattern server. That means the public server on the Amazon cloud, or at Indiana University Supercompute Cluster, for example, or if you run your own GenePattern server, you can connect to that as well. To select an analysis, there's a browser window which launches from within the notebook that displays all of the available analyses. The analysis cell is the main addition to the notebook interface. This is where the user enters input datasets and parameters, as in a web form. Once you launch a job, you will see a job cell that displays the status of the job and gives you access to result files. We've made GenePattern analyses seamless with the Jupyter Notebook system. This means that you can use a Python variable as an input to a GenePattern analysis, and you can load the result of a GenePattern analysis directly into a Pandas data frame or Python variable. To further increase ease of use by non-programmers, we've developed a user-friendly interface to Python functions so that notebook developers can provide a clean interface to their functions that expose only the parameters that are required to run them. We've developed what we call the UI Builder. This allows notebook developers to add one decorator to their functions, which describes the inputs that they want the user to be aware of, and to then automatically create a web-based interface for that cell. This hides the complexity of the code and makes the functionality much clearer. Developers can specify file inputs, drop-down lists, and can tailor the interface in many ways to the specifics of their method. You can click a button to toggle back and forth between the code and UI Builder views, and any changes to the code are automatically updated in the UI view. You saw earlier that even just creating text in a notebook requires a programming type perspective. We've addressed this as well. We have added a rich text editor that lets you add text as you would any word processor. This is an added feature and doesn't replace the other ways of adding text. This feature makes it easy to add elements that are common in scientific literature, but difficult or impossible using the native Markdown language supported by Jupyter, such as superscripts, subscripts, and special characters. You can easily toggle between rich text editor, HTML, and the final rendered text view. Now let's look at how we use these features to create the collection of single-cell notebooks that are available on our online workspace, notebook.genepattern.org. Using the components of the GenePattern ecosystem, we have wrapped several tools in modules and notebooks that span a wide array of popular single-cell analyses, such as data normalization, clustering, differential expression, pseudotime analysis, and batch correction. I will describe our implementation of the Seurat team's introductory vignette, the CoGAPs notebook for identifying patterns within high-dimensional gene expression data, the Stream notebook for computing pseudotime-based representation of biological processes, and the Kanos notebook for aligning and normalizing the expression data of cells that encompass similar biology but come from different experiments. A number of single-cell software tools provide analysis workflows in which users examine intermediate results at each step as part of an interactive process. The developers of the Seurat toolkit in the Raul Satija lab authored a tutorial walking users through downloading, pre-processing, clustering, and performing differential expression on a dataset of peripheral blood mononuclear cells. We adapted this tutorial to the GenePattern notebook environment. Here you can see an example of a module that can load many of the single-cell file types, removing the need for users to configure their analyses for different formats. The cell loads the single-cell data in any of these formats and displays a visual representation of various common quality control metrics used for filtering single-cell data. We have also published a notebook and series of modules designed to run the CoGAPs analysis from the lab of Ilana Fertig at Johns Hopkins for finding biologically relevant patterns in single-cell gene expression data. CoGAPs uses non-negative matrix factorization to reduce the high dimensionality of the data and cut through noise to find patterns in the data that may indicate the presence of certain cell types or the activity of certain pathways or processes. As we can see in this example on the left, pattern 4 encompasses a specific transcriptional signature exclusive to the natural killer cells in this particular dataset. On the right, we have a brief view of part of the CoGAPs GenePattern notebook. The notebook is rendering visualizations of various dimensionality reduction techniques common to single-cell analysis. The CoGAPs workflow is implemented in different GenePattern modules embedded throughout the notebook. Each module has configuration options for the figures and instructions above it to guide users in selecting those parameters. When executed, these modules send their data and parameters to the GenePattern cloud server. This is crucial because some CoGAP steps can take several hours. While these modules are running, users can close their notebook and the module results will be waiting when they return. We have also implemented a pseudotime method called Stream from the lab of Luca Pinello using GenePattern modules and notebooks. Pseudotime analyses use techniques such as dimensionality reduction and applications of graph theory to identify these processes in the data and order the cells along a pseudotime trajectory. As a note, in this ordering, the distance between one cell and another does not indicate a difference in terms of real units of time. It only indicates which cell comes after another in the sequence of a specific biological process. Stream uses a method called modified locally linear embedding to build pseudotime trajectories. On the left, you can see an example of Stream's intuitive pseudotime representations, which the authors call subway plots. In these plots, you can see both the different branches of the trajectory as well as the density of cells along the trajectory. The video on the right shows how the image files generated by one of the Stream modules can be downloaded and displayed in the notebook using just a couple of clicks. As the volume of publicly available single-cell data has increased in recent years, many researchers are focused on similar topics, such as immune cell and tumor interactions. There's a growing interest in being able to harmonize or align different data sets that were collected in different labs or slightly different experimental conditions, but that still, in theory, describe similar biology or are expected to contain similar populations of cells. Kanos, from the lab of Peter Karchenko, is one such algorithm that uses a clustering and graph-based approach to link populations of similar cells from different experiments. We will soon make available a notebook that encompasses this workflow, utilizing GenePattern modules to perform the heavy lifting in the cloud while enabling users to interact with the results and configure and generate the plots in real time. These notebooks and many others are available on the GenePattern Notebook Workspace, notebook.genepattern.org. This is a free resource where you can create your own notebooks and run them using only a web browser. All of the popular scientific Python libraries are pre-installed, and if there's a package you need, you can install it into your own space. You can share notebooks with collaborators or with the public. By making a notebook public, you can embed a link to it in a publication where others can easily open it and adapt it to their own purposes. We have created tutorial notebooks that teach the functions of the environment, as well as the collection of analysis notebooks I described previously, and other notebooks that feature machine learning, differential expression, and other important bioinformatics analyses. For those of you who are computational biologists, all of the functionality I described to run and access gene pattern analyses is available through our Python library, and the code for all components of the gene pattern ecosystem is open source, available on GitHub. If you do want to install GenePattern Notebook locally as an extension to your own Jupyter system, you can either grab our Docker image, which contains a complete Jupyter plus GenePattern installation, or you can install using the pip or conda package managers. We also have a Docker image for the entire notebook repository, so people can host their own GenePattern notebook repositories. You don't need to have your own GenePattern server in order to run the GenePattern Jupyter extension. It will connect to any GenePattern server on the internet. I'd like to acknowledge the GenePattern notebook developers and computational biologists in the Messerov lab and all of our research collaborators, and I'll be glad to take any questions. Thank you.